Hi everyone, welcome to this M2D2 Talks. Today we have two speakers, Brian Trippe and Jason Yim, and the talk is about diffusion probabilistic modeling of 3D protein backbone for the motif-scaffolding problem. A bit about both speakers before we start. Brian Trippe is a postdoctoral fellow at the Columbia University in the Department of Statistics and a visiting researcher at the Institute of Protein Design at the University of Washington. Previously, he completed his PhD at MIT, where his thesis centered on Bayesian statistical methodology for high-dimensional linear modeling with application in computational biology. Jason Yim is a PhD student at MIT, advised by Regina Basile and Tommy Giacola. Previously, he worked as a research engineer at DeepMind. His interest lies in probabilistic and geometric machine learning with application to design problems, in which he collaborated at the Institute of Protein Design as well. Thank you both, Brian and Jason, for our invitation to present your work here, and looking forward to the talk. For everyone in the audience, Brian and Jason agreed to take some clarifying questions during the talk. You can raise your hand or type in the chat. And for any discussion related question, we can wait at the end of the talk. Thank you, everyone. The floor is yours, guys.
Great. Thanks, Prudencio. Yeah, so as Prudencio was saying, we're today going to be talking about the motif scaffolding problem. And in this talk, we're going to argue for a probabilistic machine learning approach to this problem and describe how diffusion probabilistic models can be used to attack it. So as a bit of background, like proteins are the biomolecules that do just about everything interesting and exciting that happens in biology, from accelerating like chemical reactions that are needed for digesting food to generating motion in our muscles. And already harnessing natural proteins has been like truly transformative to bioengineering and synthetic biology, thinking of examples like the CRISPR-Cas systems and the green fluorescent protein. But in the context of computational protein design, we ask, can we do even better than this? And can we create proteins with either the same functionalities, but better suited to the kind of application needs for them, or proteins with new functionalities that don't exist in nature? The starting point today is that for a host of protein design problems, and for proteins in general, like their biochemical function is typically imparted by just a small subset of residues that we can refer to as a functional motif. Kind of inspired by this, then, a common workflow for computational protein design involves a two-step procedure, where in the first stage, which we can refer to as motif identification, we figure out which this subset of residues are, right? So for instance, maybe we have two different segments of a binding interface from different native proteins. And together these form what would make a single, like, very energetically favorable interaction. And then in the second stage, once we have this motif, we desire to scaffold it. So that's building the rest of the protein around it that can stably hold that scaffold and this functional motif in place. So this problem can have a wide variety of different instantiations with motifs with various functions coming from different sources. So like in this first example, right, it was talking about protein-protein interactions, where like in addition to potentially getting this from like a native protein interaction, we might have this from fragment docking calculations. But farther afield, we could also be interested in and are interested in designing enzymes. So in that case, from like quantum chemistry calculations, we can figure out exactly maybe where a subset of residues would have to be to stabilize the transition state of a reaction, or in a kind of similar application, maybe we want to stabilize some metal and have a metal binding site as the motif of interest. And then as the third example, for vaccine design, sometimes we'll have a fragment of a viral protein, and it's desirable to put that in some new scaffold that displays this perhaps a conserved viral epitope to the immune system, so that like when it's introduced as a drug, it can help in mounting an immune response sensitive to that particular viral piece. And while there's a lot of exciting work kind of on this bottom line of coming up with different functional motifs that could perform these different functions, in this talk, we're going to be focusing on just the second stage of this pipeline. So assuming that we're provided with a functional motif parameterized by like three-dimensional coordinates of where the residues are, and then the goal is to come up with ultimately the sequence of a protein that will fold up and contain that. So what makes this problem challenging? It's been said that in this like post-AlphaFold, like post-structure world, that protein design is now just essentially like a game of guess and check, where hypothetically we could solve like these sorts of problems around protein design by simply just checking many different possible sequences, predicting their structure in silico, and just asking like, does this predicted structure contain this functional motif? And then if it did, then we would be done. So if this were the case, like a naive approach could be just to guess a bunch of random sequences. But what's the problem with this? So the clear problem is that, even if we're just interested in only 100 residue long proteins, since there are 20 different amino acids, we have this like astronomical number of possible sequences and therefore also structures. And we can think about visualizing the space of possible structures in this kind of blank, wide open, two-dimensional space, which is, of which we can think of visualizing the structures with the motif as this near infinitesimally small green sliver. So if we're just randomly guessing, we're not going to be hitting this green line. And an alternative approach that in some settings has been successfully used is saying, like, let me search amongst like existing natural structures, searching through and finding some that might contain this motif. And we can think of this as kind of exploring this sparse subset of samples of existing structures. But this again has this limitation that like because like the segment of the space that contains the motif is so small, this is going to be too sparse, and for many interesting motifs, like we'll be far from having something that will work. There's been an explosion of machine learning work recently into protein design, some of which could is somewhat applicable to this problem. And we'll get into some of the details of this related work later on. But at least as a kind of coarse generalization of this, the existing tools struggle to give significant diversity in the structures that they produce, and can have high and in addition, highly unpredictable computational cost. So it can be intractable and unreliable to use in practice. To address these challenges, our approach is going to involve two separate stages. So the first is going to be looking at this whole world of many different natural structures and learning a probabilistic model that I'll call p theta of x that assigns a density to different possible structures. And I'm representing that here in this plot with this color bar, where higher, the brighter color is corresponding to higher density. And then in the second step, we're going to think about partitioning structures into these two segments of the motif and the scaffold, and then doing protein design and design of scaffolds as sampling from the conditional distributions implied by this model p theta of x. So, and I'll write that as sampling x sub s, x for the scaffold, from that conditional of p x sub s given x sub m. And what we find is that diffusion generative models and sequential Monte Carlo end up being key algorithmic tools for solving this problem. And together, what we'll show is that these methods have the potential to build long and diverse scaffolds, really provide a wealth of different possible solutions to this motif scaffolding problem for a given motif. As a roadmap for the rest of the talk, because diffusion models are kind of a centerpiece of the algorithmic like segment of this work, we're going to begin with some background on diffusion probabilistic models. Then we're going to discuss how we adapt this approach to this special case of modeling a distribution over protein backbones, and say a little bit about like the model's performance and its limitations. Then we'll move on to the second stage of our design pipeline, so sampling the scaffolds given the motif. And for this, we develop an algorithm that we call SMCDiff, for sequential Monte Carlo in diffusion models. And in this segment of the talk, we'll first explain why we think conditional sampling is really the right way to approach this problem relative to naive in painting, before going into the details of this algorithm that we've proven with theory is asymptotically exact in terms of the quality of the conditional samples that it provides in a large computation limit. Then finally, we'll talk about some limitations of this whole pipeline and discuss related work and in some future directions. With that, I'll pass it off to Jason.
Okay, so as Brian said, I would like to first go over kind of some background on diffusion models, and then we'll discuss kind of how we've adapted diffusion models to work on protein backbones. A lot of these slides and figures were borrowed from this tutorial that was given back in CVPR this year. The title of that tutorial is down there, and I highly recommend checking it out to learn more about this class of models. So if you kind of looked on Twitter recently or even the news, you'll kind of seen images such as these that are basically generated from artificial intelligence. And so the technique behind these models are kind of from diffusion models, where they're able to generate these really high quality, high resolution images, also kind of controlled images where you can give it these text prompts, and the neural networks will generate these images that kind of closely match what the text prompt was trying to semantically say. And so these are two models, DALL-E 2 and ImageGen, that came from OpenAI and Google recently, but these are kind of already outdated, as in there's a ton of better diffusion models out there that already also work on audio as well as videos as well. So kind of diffusion model is kind of becoming the dominant architecture in the generative modeling space very quickly. We're going to start with some background of diffusion models. The general idea of how diffusion models works is that there are two processes. The first is what we call the diffusion process, where you kind of start from the data. In this case, is an image of a cat from the left, and you iteratively add a little bit of noise until it goes to the end, where it becomes something that's just pure noise and nothing of the cat is in the image anymore. The second portion is what we call the reverse denoising process, where you kind of start from a noise and you go backwards by removing the noise that was originally added. And this is what we kind of term as a generative process of generating what the original data was. You can imagine that what general models allows you to do, you can just sample any noise sample from like a standard Gaussian, and you can generate like any sort of data and like hopefully have it be highly diverse and get very good samples out of it. So more detail on how the forward process works is that first, we define the total number of steps, which is given by this capital T, and x0 is going to be what we say is like the true data distribution. And each transition of going from kind of t-1 to the next time step is essentially a Gaussian distribution. And that's written here as q of going to the next time step t, given the previous time step t-1. And you can see that this Gaussian distribution is parameterized in terms of the previous time step xt-1, as well as this parameter is called beta t. This beta t kind of controls how much noise is added at each step. And so from the original paper, one of the original papers, from Jonathan Ho, was to kind of define the first beta t to be like very small, 0.0001, and then the last beta t of going from the penultimate time step to the last one is 0.2. And you kind of have this in an increasing step so that you're adding more and more noise each time. And then 1 minus this beta t is kind of how much signal is kept at each time step. And from the form of this Gaussian distribution of this transition, you can see that this 1 minus beta t is kind of controlling how much signal from the previous time step is kept. And so we want to set up this beta t such that the last time step ends up becoming a true Gaussian, so we can sample in the reverse process easily. So because you're kind of conditioning on the previous time step, we can break this up into a series of these kind of conditionals. So kind of the full distribution across all the time steps t would be kind of too intractable to kind of capture fully. So we kind of have this Markovian assumption on it such that at a certain time step, it's only dependent on the previous random variable of xt-1. So to train this model, you want to train a model such that it learns every xt very well. However, this is kind of expensive for large t's, where if you want to kind of sample t equals to 100 something, you're going to have to sample for 100 steps to get to that point. If you want to like sample from that distribution and then use that to train. So instead, what we actually care about is what we call the marginal forward distribution, where kind of, as I said, to sample from q of xt at t, instead of doing it in O of t time, you want to do it in constant time to be able to sample from that point of the diffusion kind of immediately. So to do this, we'll first define this auxiliary variable, bar alpha t, where it's just kind of the cumulative product of all the 1 minus beta t's up to kind of that point here. And then we're going to define what the marginal distribution of q of xt given x naught is going to be by this Gaussian distribution, where this alpha t kind of tells us kind of how much signal has been retained up to this time step. And then kind of 1 minus this bar alpha t is how much noise has been injected at this point. And a reason that this is possible is because we kind of fixed the amount of noise that was given at each time step. So this is kind of always analytically can be written down and tractable to sample from. So kind of like a kind of more technical detail is that now that we have this diffusion kernel, we can now sample from this unconditional diffusion kernel, which is q of xt. So you can kind of break it up into like a form of an expectation where you kind of have this integral over the x naughts here. And if you kind of assume that the x naught is a uniform, then you can sample from, you can get xt just given this x naught. And so that's what the form on the bottom right-hand side here, which is where given the original data point, you just sample from a standard Gaussian, this epsilon, and then scale both of those terms depending on where you want to sample from this t in the diffusion process. So in the reverse process is just the other way around, where you instead start from a standard normal distribution at the very end, and you're going backwards towards the data distribution. So this is written here as p of x capital T of being starting from a standard normal. And the goal is that in each step, as we go in the backward process, is that we want this to kind of match what the forward distribution was. So you're iteratively removing some of the noise that was added in the original diffusion process. But because when we go backwards, we don't know what the x naught is, that's why to train this neural network that can kind of recover what the true data was, or how much noise was added in the original process. So that's why kind of this reverse process of xt minus 1 given x naught, we have this mu theta, which we're going to train or learn using a neural network. So again, instead of kind of having the model learn kind of this whole distribution, which would be intractable, we kind of also has Markovian assumptions such that it's broken up into each of the individual terms of how to go back from each time step, kind of an iterative process. So sampling in this way is actually very expensive. There's no way to kind of do a marginal of jumping all the way back to x naught in a single shot. And so these t, what people have found is that you want to make this very large so that you can kind of make the model only work a little bit each time it goes backwards. And so t is on the range from 100 to 1,000, depending on kind of which data domain or problem that you're working in. But obviously kind of 1,000 steps for a very expensive model is very slow. Therefore, it's kind of currently an active area of research to kind of speed up how one can sample from these models very quickly. So to kind of actually learn this reverse process, we're going to talk a little bit about optimization. So again, this is the reverse transition that I want to learn with the neural network. And if you recall, this is the original form of how we're sampling from the forward distribution. And if you kind of play around with the terms a bit, you can write down what this mu theta is here. And it is closely related to x naught. So just in terms of like time, we're not going to kind of derive it here. The tutorial has more details on how to get here. But this mu theta can be written as this form where you kind of have the beta t's and the alpha t's and this xt, which the model sees. But really what is unknown when you do this reverse process is how much noise is added. And so that's given as this epsilon theta here, which is what you want the model to essentially learn in the reverse process. And so again, from the seminal Johnson Ho paper of diffusion models, he showed that you can reparameterize this such that all the model has to learn is epsilon data, given what the true noise was in the forward diffusion process. So this is a very simple loss that works really well in practice, that the model can be easily optimized for. And that's why in the literature of diffusion models, people kind of refer to this as a noise prediction model to learn the noise that was added and how to remove it when you go in the backwards direction. So that's kind of technical details that'll be important when Brian goes over the sequential Monte Carlo algorithm. Some intuition of why diffusion models works is that one is that they kind of remove this need for an encoder, as in this diffusion process is kind of this encoder already that encodes what the original data was from the left-hand side into this right-hand side. And so that's kind of what this picture is depicting, except in the continuous time where you kind of take the number of t's to infinity. So you can kind of draw it out that there's this kind of Brownian motion happening starting from the data distribution all the way up to this standard Gaussian at the very end here. And so kind of compared to VAEs where you have to train the encoder and the decoder, here you just have to train a decoder of going to the backward direction. And then the decoder, as compared to GANs, is kind of easy to train. As in the previous slide, you just have to learn kind of how to predict this epsilon, which is just a standard normal variable. So it's much easier than GANs, which have been shown to be kind of very unstable to learn when you do kind of adversarial type training. So kind of why do we think diffusion was good for proteins? One is that they allow us to model directly in 3D space instead of something such as histograms, which previous machine learning techniques have been based on for protein design. And by doing this, you can kind of condition on parts of 3D, which is perfect for Motif-scaffolding. And also kind of gives a better inductive bias to the model because it's directly predicting things in 3D space rather than having to kind of project back and forth between 3D and 2D distances, et cetera. And already kind of there's been explosion to diffusion models for molecular generation. So kind of one of the first was by Hoojboom et al. From Max Welling's group, where they show state-of-the-art success in generating small molecules for things like drug design. And then there's also another paper that kind of doing diffusion models for point cloud generations, where they were able to generate kind of high fidelity point clouds that weren't previously been able to do before. So kind of when we want to apply this to proteins, because you can just treat Gaussians in like a multivariate Gaussian in 3D space, it's actually very easy to inject Gaussian noise into the 3D coordinates of a protein backbone. And so here kind of instead of like an image in 2D, we now have Gaussian point cloud in 3D. And then at the very end is kind of what just a 3D point cloud would look like for the C alpha coordinates of a protein. And that's what this GIF is showing of going backwards or is it going backwards? Yeah, it's good. Yeah, so going backwards from like the noise distribution to the data distribution of like, how it would look like. So in this problem, we just kind of concern ourselves with just the C alpha coordinates of a protein, but obviously that's not the full dimensionality of protein or kind of full representation. In future work, we want to incorporate things such as the all-atom representation of a protein, which would require modeling the rigid bodies as done in AlphaFold, also incorporating the sequence. But kind of one reason that we went down this route is that there's already a ton of tools out there to kind of recover the sequence as well as generate the full atom. And because the theory of how to do diffusion on just like a point cloud is already there, so it kind of paved the way for us just to focus on the application of this problem rather than trying to develop the theory of the full protein representation. So for the model detail, when you do this 3D backbone, there are some important things one needs to be mindful of. The first is to be translation invariant. So that can be easily achieved by zero centering your 3D backbone point clouds. So just not like at point 1,000, 1,000, 1,000 or something. And so by always zero centering, you don't have to worry about translations at all. And second, compared to small molecules, which you can get away with kind of sparse graphs, we found that it's very important to treat the protein as a fully connected graph such that every residue can see every other residue in this graph structure. And that's because these long distance interactions are very important in proteins when it folds up. Another thing is that it was important to break the permutation equivariance because proteins have a chain-like structure that needs to be maintained or kind of respected in a realistic protein. So we also incorporate the sequence position indices, very similar to kind of how transformers work, of each residue into the model. And another thing is that the model wants to know kind of what time step is as well, because every time step is a different degree of how much noise is added. So by letting the model know the time step, it can actually kind of control how much it scales the protein by when it predicts the noise. So kind of at the heart of doing any sort of geometric deep learning is this notion of equivariance. And so E3 equivariance refers to Euclidean transformations. You want to be equivariant to transformations in 3D, which encapsulates translations, rotations, and reflections. And as I mentioned, because we're zero centering, translation is already taken care of. So what you really care about is reflections. Sorry, not reflections, but rotations. And so that's given by this goal here, where you want the probability of your point cloud x0 to be the same as if that point cloud is rotated by this rotation matrix R that's taken from the SO3 group. So kind of to write down in words, kind of what the desiderata is for this model is that starting from an invariant distribution, you want to use an equivariant noise prediction model such that you get equivariant samples. So that's kind of hard to kind of take in words. So there's this diagram on the right-hand side to show this, where that if you just rotate the starting sample from your noise distribution, that's this xt, big T here, and you rotate it 90 degrees, you want your sample to essentially be the same thing, but just rotate it as well. So the model doesn't have to expend more model expressivity to kind of learn kind of how to deal with this rotated symmetry of the starting distribution. And so this is kind of the ultimate kind of thing that I want this model to have. And this is kind of nicely already done for us by previous work by GeoDiff, by Zhu et al. That kind of proved that if you have this equivariant noise network and an invariant distribution, that yeah, you get equivariant samples here. So to achieve this, we use equivariant graph neural networks that have also kind of become quite popular of achieving this E3 type symmetry by Satoros and in Welling's group, Max Welling's group. And this has the property that it's an equivariant neural network. So kind of written down there that if you rotate the input of this like Rx, that it's going to be the same thing as if you just rotated the output of the unrotated input. So this leads to equivariant noise predictions. So kind of one thing that's kind of an elephant in the room is that this is E3 and proteins are not invariant to reflections, as in if you reflect the protein, then it's going to have different properties, different biophysical properties. But like in this problem, because we're just dealing with the C alpha coordinates, that's like a limitation that we accepted to work in this framework. But it's a next step to kind of work in the SC3 equivariant framework. So another thing that we were also very concerned about in this problem was like how to actually evaluate your designs. Generative models are kind of notorious for having kind of very opaque metrics that are susceptible to a lot of biases. And so we wanted to kind of think about how do you evaluate these proteins properly. And so we kind of came up with something we call designability. Well, it was already kind of done in David Baker's lab. And so we kind of just made it a little bit more formal here, that a 3D structure, we determined it as designable if one could design a sequence that it could fold into. And so because it's expensive to run a lot of experiments to fold every single design you have, what we use is a very good folding model, such as AlphaFold, to kind of see if the predicted structure from AlphaFold would match what the original sample design is from your generative model. And so that's kind of what this diagram on the right-hand side shows, where the sample backbone would come from our diffusion model. We would have some sort of sequence design method to generate the sequence, and then run that through a structure prediction model such as AlphaFold, and then compare that with the original backbone. And then we would use something called a TM score, which is very similar to kind of RMSD of the C alpha positions. But TM score is different, as in it has a normalization factor to make it not so sensitive to the length of the protein. And so, and the nice thing about the TM score is that it's between 0 and 1. And people have kind of empirically said that anything above 0.5 roughly indicates the same structure between the two proteins. So before I go on, any questions about this designability criterion? Okay, yeah, feel free to interrupt me at any point. Oh yeah, so I guess yeah, one thing
I had a question. How are you going to train the sequence generator here?
Yes, so sequence generator could be trained end to end, but what we decided to do was use kind of off-the-shelf methods for the sequence design. So if you've kind of seen, there's been a lot of sequence design methods out there from Facebook. They have an inverse folding method of going from a structure to a sequence. There's also from the David Baker group called Protein MPNN. That's a message passing neural network that's also very good at going from the structure to the sequence. We don't train these, we kind of just take them as pre-trained models. I think this is also kind of interesting as well to kind of have these two separate models that if they agree together, although they're kind of separately trained, that's kind of a good indicator that these two independent oracles kind of give a coherent like structure and sequence relationship at the end. And also the same thing for the structure prediction, as in we just take the pre-trained AlphaFold model as a way to validate our neural network. Okay, yeah, sorry. So this is kind of actually what the next slide was about. So we take the protein MPNN neural network that was already pre-trained for us, and we only give it the C alpha distances as opposed to the frames that it was originally trained on. And then for the prediction network, we use AlphaFold, but we don't use MSAs or templates because we want to design truly de novo proteins. So we don't want to mimic or kind of bias AlphaFold to kind of give high confidence for things that are outside of, or yeah, only give high confidence for things that are already observed in nature. And I guess I'll just put out there that obviously there's a lot of methods out there for structure design, sequence design, like from Facebook. There's also a lot for structure prediction. There's things like OmegaFold that only don't require MSA at all. They just require you don't input the sequence. So because our preprint kind of came out after, before those papers, it's something that I hope a lot of people, or people have already started to explore kind of the designability criterion using these other structure prediction and sequence design methods. So just like an idea of what this metric kind of entails is that we noticed that if given the backbone design that's in this first column and kind of what AlphaFold predicts after the sequence design step, if there's a very high agreement between them given by 0.9 SCTM. Oh, I forgot to mention that SC stands for self-consistency, to mean that it's consistent with kind of what AlphaFold predicts. Yeah, so we noticed that if the relationship between the two is very accurate, about 0.9, it usually means that we're kind of just recapitulating something that's coming out of PDB. So kind of because we're generating something that's very similar in PDB, and because AlphaFold was trained on PDB, like AlphaFold gives it a high score, and so we're not doing anything interesting here. What's more interesting if you kind of go into this region of SCTM kind of in the 0.5 region, where AlphaFold is kind of like not so sure what the structure is because it's never been trained on it, but it kind of still gets like a structure that looks kind of reasonable, as like the beta sheets in the front are not perfect, but there's definitely some alpha helices in the back that are being formed. This does not have like a very closely matching thing in PDB. So this is kind of an indicator that the model, even after these filtering criterions from AlphaFold and protein MPNN is kind of generating something that's kind of novel and not found in PDB. So it's kind of 0.5 region is kind of the sweet spot of what, or maybe 0.5 to 0.7 is kind of sweet spot of what you want to look for in generative modeling. And in SCTM, anything below that is kind of just crap. It doesn't form anything that's useful here. Okay, also realized we're out of time, so I'm going to go a little bit faster. So to start off for this the neural network, we trained it with just like short proteins. We know it's having a little bit of trouble going up to things that are greater than 128 residues. That's definitely an area of future work where we want to kind of generate longer proteins. But so because of this this, like what we saw only sticking up to 128, we kind of forced ourselves to only work with about 4,000 proteins from the PDB to train on initially. And then, yeah, just as a sampling pipeline, again, you take the diffusion model, you generate the backbone, use protein MPNN to get the sequence, then AlphaFold to get the all atom structure of it. And using this pipeline, after training it, we kind of looked at how many of them are designable. And so we see that kind of to the left of this vertical line is like a histogram. Sorry, this is a histogram of the proteins that were sampled. And the blue are like all the short proteins that are between 50 and 70 residues, and then the long ones are between 71 and 128. And the things that are to the left of this are determined not designable because they're lower than 0.5 SCTM. So the most of the things from our neural network are not designable, but there's still a certain amount of things that are designable past that, about 12% of the designs, which is still very higher than kind of conventional protein design methods. And so we also mentioned that a big limitation of our method is that it's not SC3 equivariant, it's only E3 equivariant. So there are still reflections that happen within the protein. And so we're kind of marking that here as these, these X's and the O's that are also colored. And another thing was that we made a scatter plot of comparing our designs by doing a pairwise SCTM of everything that was in the training set. So I always get them mixed up, but so we want things that are very different than our training set. So kind of above this line, but also things that are designable. So to the right of this line. So everything in the top right-hand quadrant is kind of what we want our model to generate here. But close to 0.5 as possible for the comparison to training sets, such that it's different than a training set. And so we took one of these that's in the red box, and we looked at it, and we saw that the model's generating things that are designable that AlphaFold can generate, but also it has nothing that's similar in the training set. So it's generating novel folds that weren't seen before in PEB. Prudential, you had a question?
Yes, a small clarifying question. Why do you limit yourself to 128 residue?
Yeah, so we noticed that if you go up to 256, it was kind of tough to train these models. One thing was like we had some trouble scaling EGNN, but kind of in like what we're working on now is kind of go up to longer proteins essentially.
Okay, so it wasn't because of the diffusion, but rather because of the EGNN for the generation, I guess.
Yeah, I think the biggest effect is because we're using E3 neural networks. So this chirality issue becomes much more prominent. As in, the more residues you have, there's a higher probability of a residue having a bad chirality in there. So this probability of the incorrect chirality exponentially gets higher as you go up to these larger proteins. So that was the biggest limitation. And I think kind of midway through the project, we kind of regretted that. But it's something that we're definitely fixing in our next paper.
Thanks.
Okay, yeah, so kind of as a good segue into that, like these are some specific failure modes to show kind of the limitations of our model, which is that left-handed helices are definitely possible because it doesn't know about the reflections that are happening within the residues. And the other thing is that we still know that there are some chain breaks. So because diffusion is quite a noisy process and Gaussian noise isn't like a geometrically inspired noise that you can add into a protein. So when you do like Gaussian noise into a C alpha backbone, it's obviously going to break the bonds that should have been there in the protein. So like another very interesting area of research is maybe kind of thinking about a new type of forward noising process that'll maybe respect the bond geometry better. But because of the inherent noise that's in this diffusion model, there are definitely some chain breaks that the model still can't perfectly learn to respect. And for sample interpolations, this is where we're just kind of interpolating the noise between two randomly sampled proteins. And you can notice that it's a smooth translation or transition between kind of two different folds to show that the model is learning something interesting in the noise distribution. Okay. Yeah, sorry, I'll give it to you, Brian.
Yeah. Thanks, Jason. Great, yeah, so now going to move on to the second stage of this design pipeline, which is taking the learned protiff model and then constructing scaffolds around motifs by conditionally sampling from it with an algorithm that we call SMCDiff. So as kind of setting the stage for this, in addition to text conditional image generation, diffusion models have also shown really remarkable performance on text conditional image in painting. So for example, in this framework Glide from OpenAI, we can see in this example, you can take this painting of a girl with a dog and then mask out the section with the dog and then in-paint it, conditional on the text, a girl hugging a corgi on a pedestal. And you get this like really remarkable looking image where, yeah, I mean, this dog has been replaced with a corgi. And while this is, I think, really incredible, if you look like really very closely at the in-painting, particularly at the boundaries of it, in this case and also in a host of others, you see that you do get some artifacts and edge effects. So for example, here, if we look at the girl's pearl necklace, you can see it's present on the left side, but after in-painting, it kind of disappears on the right. It's kind of gone from this continuity is gone. And similarly, below this, the dog's feet at this drawer, there are some white splotches that it's not really clear like what these could be coming from. And while this really, I mean, might be imperceptible from a first glance when you're looking at images, in the context of protein design, these sorts of small errors are like crucially important because they break designability, right? If we're off by even just like a nanometer, then this will leave us with a molecule that couldn't even exist in nature. So, I mean, a first impulse looking at this, I think, is to ask, like, what is going wrong here? Like, why is this kind of failing in these ways? But I'd argue that really the more relevant question to be asking is, why is this even working at all in the first place? And the kind of working hypothesis for this is that the successful in-painting algorithms are doing approximate conditional sampling from these diffusion models, and that it's the approximation error that's leading to these artifacts. So why is conditional sampling really the right way to think about this problem in our mind? To answer this question, let's return back to the schematic that we had from the beginning and try to think about this problem probabilistically. So starting off with kind of this assumption, now that we're armed with this language of designability, if we make this assumption that the model that we fit, represented by this heat map, is going to have positive density only for structures that are designable, then this will kind of lend us a way of thinking about this scaffolding problem more formally. So getting a little bit further, right, analogous to how we divided our image into like the mask portion to be in-painted, here we're dividing our structures into a motif in blue and the scaffold in red, where the motif now is what we're going to be masking out. In mapping this onto the schematic, we can think of like dividing this space of structures into the scaffold on the x-axis and then the motif on the vertical axis. So our rationale then is that if for a given motif of interest, xm star, we have that when we look at this density restricted to this sliver and kind of integrate over it, average across it, and consider this marginal distribution over motifs that could exist. If that marginal distribution, so just P of xm stars is greater than 0 at this desired motif, then the conditional distributions implied by this model are going to be perfectly well defined. And under this first assumption at the top, any scaffold that we sample from it will give us a designable structure as a whole. So yeah, thinking now, like we can represent this potentially complicated conditional in green up at the top. And our goal is to think about looking at these densities, these regions of high density in this conditional distribution. So for example, this like scaffold in red over here could be one solution, but in general, there might be a diverse set of possible scaffolds that would complete this that have high density here. So just emphasizing this again, with a good enough conditional model that's satisfying this designability assumption, motif scaffolding is really just equating to conditional sampling. But the challenge then is, how do we actually obtain conditional samples from this model? I'm assuming that we have a good unconditional model. And for that, yeah, I'm going to talk about our solution to this with sequential Monte Carlo diffusion. So yeah, as a starting point, recall that diffusion models only really directly give us a way to do unconditional sampling by going kind of one step back at a time for the entire structure, given the entire structure just one step further forward in the forward diffusion process. But this isn't really what we need for conditional sampling. If we wanted to do this exactly, kind of a more desirable approach would be to build up the scaffold segment across time, this reverse process, but conditioning instead of on the motif at each of these later steps, conditioning on the motif at time step 0, right? So that we're looking all the way forward and capturing this totally unnoised motif instead. And while this procedure instead would give us exact conditional samples, it's going to be computationally intractable. because again, like we only have access to these particular conditional distributions with the diffusion model. So as a tractable alternative, which is what's used in these state-of-the-art image in-painting algorithms, is something that I'm going to refer to as this replacement approach, which broadly is defined by, or I guess I should say specifically is defined by first applying the forward process to the motif. So this is up on the top and noising that all the way to time big T. And then in a second step, doing this reverse replacement sampling, where we're only sampling the scaffold and slotting in this forward diffused motif at each time step. And while this kind of keeps things more or less in distribution relative to perhaps just keeping the motif fixed in place at each step, and this is kind of an approximation error, an approximation algorithm that leads to error that in our experiments we found gives rise to some two chain breaks. So thinking about these two options at like one end of the spectrum of this replacement method, which I've argued is inexact but tractable, and this exact but intractable conditional sampling procedure, we ask, is there something in between that maybe could allow us to trade off and get somewhere in the middle? And our proposal for doing this is going to be exactly getting at this kind of in-between space, which is to start with the replacement method, but try to look ahead by a few more steps. In particular at time t and t-1 for the motif, in addition to just time t+1 as we had before. And what we find is that getting at these conditionals is actually tractable with a sequential Monte Carlo algorithm. So picking this conditional distribution apart just a little bit, we can see that this conditional, the scaffold given these three motif steps and the scaffold at time t+1. By Bayes' rule, we can write this as the probability of this joint distribution, looking at both the motif and the scaffold at these steps. But then we can kind of tease apart a little bit more by noticing certain aspects of the Markov structure and conditional independent structures in these graphs, in the graphical model associated with this, where we see that there's one term that's the normal diffusion model transition distribution that we can sample from, and then a second term that we can recognize as kind of a computable weighting factor. And this is what we're going to be using as the weights in this iterative sequential Monte Carlo sampling procedure. So what is this actually going to look like? So analogous to the replacement, right? We forward diffuse the motif, but instead of just reverse diffusing one scaffold, we're going to do this with a set of k-weighted scaffold particles. So at each of the steps in this forward process, we're going to be simulating k different times from proposal distributions conditioned on the associated particle at the previous step. And then we're going to assign a weight to each of these particles by looking ahead. So looking at these additional motif steps looking forward. So these are highlighted in purple to compute these once normalized weights that we're going to call W tilde. Once we have these weights, we can then resample them, resample the particles associated with these weights. In the simplest case by a multinomial like resampling, but in our paper we talk about a lower variance improved scheme relative to this. And the kind of surprising thing, if you aren't familiar with this class of algorithms, is that we're able to prove a proposition, which I'll state here informally, which is that under this case that our diffusion model the unconditional model has correctly matched the data distribution. Then in the limit that we send the number of scaffold particles k to infinity, the x naught SKs that we pull out of this algorithm at the end are going to converge in distribution to the exact conditionals of our data distribution and of the trained diffusion model. So this is significant because it allows, it provides us with a statistical computational trade-off, whereby through increasing the number of particles, which is going to linearly increase the computation that we need to use, we're able to get higher and higher precision conditional samples. But how do we evaluate this approach and this motif scaffolding in general? So for this, we're using a very similar evaluation setup to what Jason described, the conditional sampling approach, where we're like first starting with a sampled backbone where the motif is what we were given, and then we've just come up with the red. We generate a sequence for the scaffold part using protein MPNN in our paper at least, predict the structure, and then calculate the TM score to make sure that it matched the backbone that we'd come up with, and then additionally compute the recapitulation of the structure at the motif. So this motif RMSD is going to be the critical metric here. And for, yeah, I mean, again, thinking about biochemical functions that need to be performed, high resolution is very important here. So we're going to look for resolution on the order of 1 angstrom for this motif RMSD. So yeah, for full details of this, you should, yeah, please, if you're interested, take a look at our paper. But I'll discuss a little bit about 2 case studies that we've done with this, where for simplicity, we're doing this for scaffolding motifs coming from the PDB data set on which the model was actually trained. So this is something that's certainly making the problem quite a bit easier. And in large part because we know that in contrast to some protein design problems, we know that at least one solution is going to exist. Yeah, and in this case, I'll show results on 2 different examples from the PDB. One that's about 120 residues long, and the other one's about 70. And because it's been known from earlier work that the difficulty of motif scaffolding becomes harder and harder as you try to produce larger scaffolds relative to the size of the motif, we're going to test this algorithm in different variants of conditional sampling with, yeah, kind of a different size of the scaffold to be generated around it, which we're going to do by providing variable length pads around the motif. So this will be providing additional residues of these native proteins and presenting it to the network as the motif, but still only computing this motif RMSD on the motif portions highlighted in yellow here. So in these plots, right, we're going to be changing the scaffold size on the x-axis here, and then again looking at the motif RMSD. And what we see is that certainly, like as prior work has found, across the board with different sampling algorithms, we get worse performance as we move to these more challenging tasks with larger scaffolds. But we find that our conditional sampling approach, SMCDiff, with only 64 particles, is doing a much better job at providing diverse scaffolds for longer residues. So yeah, on this specific case of 5-TRV, we're getting up to 80 residue scaffolds, whereas these more naive in painting approaches, so this is the replacement method, for example, are failing beyond 50 residues. And on the bottom left, you can see 2 examples of scaffolds produced by SMCDiff with quite different topologies. We see a similar pattern when we look at the second example, although the performance breaks down a little bit faster. And we see all these approaches failing essentially beyond 40 residues of scaffold. Certainly this approach is much more of a first stage than it is a panacea for solving these problems. And as one failure case that trying to scaffold this site 5 of this neutralizing antibody found to the respiratory syncytial virus, RSV, which has been known to be difficult to scaffold, and only recently, for the first time with an alternative non-generative machine learning approach, was successfully scaffolded. So you can see some examples of this on the right side. But our approach has failed to scaffold this motif. Okay, so I guess to wind up, I was going to say a little bit about some related work on approaches for motif scaffolding and generative modeling of proteins. And something that I'll emphasize now is that we, and I think much of the field, is incredibly hopeful that generative modeling is really going to be a game changer for what machine learning can do for protein design. But at this point, we're really not there yet. And non-generative motif scaffolding approaches are very much state-of-the-art still. And an important example of something in this space, which is still a machine learning-based method, is known as a deep network hallucination of proteins, which is essentially just a local sequence search of the sequence input to AlphaFold. So this is starting with the sequence and then applying these MCMC or gradient-based updates relative to a loss on the recapitulation of the predicted motif. And this can produce some really neat looking things. This is how, this is essentially how those RSV Site 5 examples were solved in the previous slide. But this approach certainly has some serious limitations. So a first one is that it's very susceptible to adversarial examples. Again, because it's relying on AlphaFold's generalization properties. And additionally, it has compute cost on the order of hours to days with very variable success rates. So oftentimes this won't even spit something out, and you won't know if it's because you've only run it for 2 days and maybe should be spending more and more GPU time to come up with a single thing that might look reliable from in silico metrics even. A second approach is kind of a super res retraining of these structural prediction networks that I'm not going to talk about for time, but we have some more background on it, and we'll show you a reference for this paper as well. As I said before, generative models are certainly not state of the art at this point, but have been making some really massive recent strides. So the most widely used approach for generative modeling of protein structures has been on the space of distance matrices. And this is essentially just treating generation of structure in terms of these distance maps as like an image generation problem. But a limitation of this is that it relies on this like non-differentiable folding as a second step, which is kind of an awkward like downstream piece. There's also been some concurrent work on diffusion models in 3D space for protein structures. But so far, none of this other work has looked at unconditional sampling and is more focused on, like, once you have a coarse topology of a protein, how do you generate the fine-grained structure? Or if you have, say, the whole structure of an antibody and just need to build in a 10 residue loop segment, how do you do that? So kind of summarizing to put this all kind of into place, the main advantages of the approach that we have been developing so far is that it allows unconditional sampling of diverse backgrounds, of diverse backbones. And because it's in this generative framework with a fixed number of steps, we get kind of a predictable computational cost and efficient sampling of diverse scaffolds, up to 80 residues long. But I'll mention again, the really serious limitations of this method. It's very much something in active development. At this point, it's not extending beyond being able to scaffold motifs that aren't in the training set. And an additional limitation is that this method and the other ones out there for motif scaffolding require specifying as well, like the scaffold length and motif placement. So I think there's possibility for machine learning methods to offer a lot there as well. So to conclude, diffusion models enable a probabilistic approach to motif scaffolding, with ProDiff capturing this distribution over diverse backbones and SMCDiff providing accurate conditional samples. This work was done with a really great group of collaborators: Doug Tischer, Tamara Broderick, my thesis advisor, David Baker, the leader in of the Institute for Protein Design, with whom we did this work collaboratively, as well as Regina Barzilay and Tommy Yakula. If you're interested in more details, please take a look at this primary reference or email us with questions. Thank you very much.
Thank you so much, Brian and Jason, for the great presentation. It's time for questions. If you have any question, please raise your hand. There's one question in the chat by Harry that you can start with.
Sure. So Harry Guo asks, is the 128 protein sequence limit related to your assumption of the fully connected graph for the protein? How does the fully connected graph assumption affect model performance? Yeah, I think this is a good question. I think that, certainly like one of the challenges of like moving up past up to things larger than 128 residues is that like we really can't get away with completely ignoring interactions that would happen beyond that length limit. Right, I mean, it's very common in like real protein structures to have, like essential interactions occurring like even in large natural proteins, like hundreds of residues away. And if we made the network entirely blind to this, then I think it would severely limit our ability to do that. But that said, I think it's challenging to really, point down like what the error is. I mean, we haven't done this ablation of trying fitting to these larger proteins, say with like a limit on the number of neighbors being 128. But that might be a good thing to check.
Yeah, I would say like we're experimenting with ways to kind of do sparse graph representation and message passing. One inspiration for us was like AlphaFold uses a fully connected construction and how they do proteins, and kind of they model just a domain piece of it, and so they kind of show that if you just model domains, then you kind of build up the protein that way. But certainly, I think there's a lot to explore in what the best graph representation is for protein structures.
Mm-hmm. It looks like Yashua has asked a question. So Yashua says, how do you deal with variable size proteins in the diffusion model, which works on fixed size vector? Yeah, so for this essentially comes out for free with the graph neural network parameterization of the reverse diffusion model. I mean, I guess like mechanistically, right? Like we can, since our forward process is defined independently on each of the residues in the protein, like sampling the forward process is obviously relatively straightforward. And so is the like conditional score or noise that we need to predict as well. So I mean, at training time, we can just pick a protein of a random size, noise it. We have a well-defined training label, and we create a graph of the appropriate size, and just have it predict that output.
I think what Yasha's asking is like, how does the model know about the length? So this is done by the positional tokens that we give the model. So each kind of residue knows that it's on the first residue of the protein, and then it goes all the way up to like residue 128. But then that last residue might see that, oh, there's no other residue after me that I see in this fully connected graph. So the model kind of knows kind of what length it is. Another thing that we found to be kind of helpful was to kind of give the model the relative sequence lengths, so that in the edges in this fully connected graph, each residue knows kind of the sequence distance to every other residue. So that if I'm residue 10, then the edge to residue 12 would have like a 2 encoded in the edge, so that it knows that I'm 2 residues away from the residue in this graph, or like the sequence of the protein. So it's kind of like hard coded into the features of the model of the length.